So thank you all for coming today here and online. It really is an honor to introduce my friend, Dr. Luis Medina-Faull. And he's coming to us proudly from Venezuela, where he earned bachelor's and master's degrees in marine biology and marine science from the University of Oriente at 2 different campuses. And then he later earned a postgraduate diploma from the Bermuda Institute of Ocean Sciences, one of my favorite places. And the next degree from one of my, actually my favorite place, SoMAS here, PhD, under the guidance of Professor Taylor. Luis is now working in his 2nd year of what we call this prestigious fellowship, the IDEA Fellowship. And this is a position in which I believe all institutions on campus, academic programs, compete for talented individuals who demonstrate a commitment to promoting inclusion, diversity, equity, and access. And upon completion of this program, he may be invited to join us in our tenure track faculty, which is pretty exciting because I've been serving as Luis's instructor when he was a graduate student, as one of his committee members through his PhD, and now as a mentor in this program. And I can tell you he's already very ambitious. He has great ideas. He's easy to work with. And he passes the Tom Wilson test of being a genuinely good person, which is someone that we want to have work with us. He has a lot of ideas that he's expressed to me over the past few years about his basic observations that he performed in Gordon's lab and how he's taking them further. And I don't want to take any more time from him, but allow him to speak for himself and showcase his work. So it's an honor to have you here, sir.
Thank you.
I look forward to hearing from you.
Thank you, Steve, for the presentation. Okay. Thank you, Steve, for the presentation. Thank you, Laura Mary, for inviting me to the Friday seminar. I have to say that this is my 3rd time presenting my work here in this room. 1st time when I defend my dissertation. 2nd time when I apply for this position, and today. And I feel very excited today. I've been in that side of the room so many times, so many Fridays, listened to wonderful speaker, and now being here in this side of the room, I feel humble, but I feel very proud. So today I'm going to talk about the role of carbon-rich microparticles in the ocean carbon cycle and potential implication for the climate. So among all these type of carbon particles that we now can find in the ocean, I'm going to focus today on black carbon and plastics. So microplastics are any piece of plastic that can fit here in this 5 millimeter square, red square. You can see there, 5 millimeter. And for comparison, here is 1 inch. And this is very useful for me when I talk to people that are not very familiar with the metric system. So we can compare this 5 millimeter with 1 inch, and people understand what we're talking about, millimeters, micrometers. So microplastics can be grouped in 2 different categories depending on their origin. Primary microplastics are the one that intentionally includes in commercial products, like healthcare products. And secondary microplastics are the one that are the products of fragmentation processes. So imagine that old summer chair that you have in your backyard, that plastic chair that being exposed to the weather, to the sun, to the rain, to the snow, and then you sit that in that chair and you feel it's very brittle, and you touch it, it's a lot of grain in your fingers. Well, all those grains are potentially thousands or millions of microplastics that can go to the ocean. So these particles are very diverse in terms of their shape, size, color, and chemical composition. We know now there are hundreds of polymers that plastic can make of. So this made the study of the microplastic very complex. So on the other hand, black carbon are these also particles mainly consist of carbon, and they are the product of incomplete combustion of fossil fuels and vegetation, wildfires. So wildfire produce that smoke is full of black carbon. So they can have 2 different origins, pyrogenic origins from fire. So this can be charcoal or soot, and also petrogenic origins like minerals like coal, graphite, and graphene. So why I'm interested in these two types of particles? Well, first, they are mass produced. So you can see here the production of plastic from 1950 to 2015, roughly 8,000 millions of metric tons are being produced of plastic. Half of that are being disposed or discarded to the ocean, to the environment. So on the other hand, here you see black carbon, and here the pipe represents different activities in which we produce black carbon. And for example, residential biofuel, transport, and then the bubbles represent different geographic regions. So you see the abundance. So basically, these particles are everywhere, and we are witness of this because we have been sampled, for example, the Antarctic, the Arctic, the Caribbean are on here, and every one of our samples, we found microplastics and black carbon in every one of them. So in summary, they are mass produced, they are everywhere, but most importantly, they mainly consist of carbon. Between 70 and 90% of those particles are carbon. So the question is, what is the consequence of these particles in the environment? So if you think about the ocean carbon cycle, these particles are in the ocean. We know now they are in the ocean, and we know that can be potentially included in measurement of this. So the question is how these particles can affect, for example, the equipment that we use to measure different pools of the carbon cycle. How can impact the numbers that we produce with those equipment? And how can impact the interpretation of those numbers that we get for those equipment? So those are the questions that we need to understand. And also possible implication to the climate warming. So these particles are in the snow, we know now, they contribute to the snow, melting of the snow. But today, I'm gonna focus mostly in this part of these questions. So another important aspect of this is the size fraction that we are interested in. So small particles less than 300 micrometer are most likely to be included in the marine food webs, but also are most likely to be sampled with these carbon pools. For example, particular organic carbon. So these small particles are most likely to be included in those pools. So that's why we are interested in these particles, 1 micron to 300 microns. Another fact is and it's okay, depend on what size are you interested in, you use different methodologies to sample. So historically, we were using zooplankton nets to sample for microplastic, and what's happened, everything that passed through this mesh, 0.3 millimeters, 300 micrometer, escaping our detection, escaping our sample method. So we were missing a lot of those samples. So we have been able to prove through all the sampling analysis that particles smaller than 10 micrometers, 12 micrometers are the main component of this pool of plastic, of microplastics in the ocean. So we know now that these are dominant, this size of microplastics. The other aspect that is important here is not only the number of particles, it's the mass of those particles. Because when we want to understand the carbon budget, those measurements of VOC are made in terms of the mass of those carbon in the ocean. So we also need to know the mass of these small particles in order to compare, in order to include this mass of microplastics into the carbon budget. It's a matter of units. We need to know the masses to be incorporated in your budget. So, and in addition to that, we don't quite understand yet the fragmentation processes, how these particles can break down in our sampling methods with our analytical method. So it's important to also measure the mass of these particles. And I have to say, today I'm going to show you number of particles, but also I'm going to show you mass of those particles, so both of them. So how we do this? How we can measure these small particles in the ocean? So first, of course, we need to go out and sample, water samples. We do extraction, cleanup, and concentration. This cleanup is important because when you sample coastal water, even open ocean water, that water is full of things, debris, particular organic matter, things that you are not interested in. So we need to clean a little bit the sample. For that, we use hydrogen peroxide. And then we concentrate this in filters, all these particles. Hopefully, they are very clean, and we only have our beautiful microplastics. That is not always the truth, but yeah. And then we take these filters to our analytical method, and which we scan the surface of the filter. We do a scanning of the surface of the filter, and we produce maps of this surface. And then we do an interpretation of these spectral maps. So we do this part using Raman spectroscopy, and Raman offers huge advantage of comparison to other methods. For example, if Raman is telling you that that particle is plastic, it's 100% plastic. It's not room for mistake on that, on false positive or. And the other aspect is the detection of very, very, very small particles. We can go down up to 0.4, 0.3 microns. So we are talking about very small things, size of the viruses, size of the bacteria. So those are two advantages of the Raman, but also, as any analytical technique, Raman offers some limitations, which are expensive and time-consuming. And you don't want to mix those two. You don't want to mix expensive things with time-consuming things, because at the end, you're going to end up spending a lot of money in those samples. So what I'm doing now is part of my work right now is trying to decrease the time of analysis, because as a researcher, as a scientist, I cannot reduce the price of the equipment. I cannot do that. But I can decrease the time of analysis. I can reduce the time of analysis, and this is something that I'm focused right now, because I think make this method available to everyone is important. I've been reaching by a lot of community-based organizations that want to study and understand microplastic in their waters, in their communities. And when I tell them, like, hey, do you have $1,000 to analyze the samples? No. So $1,000, $700, $2,000 for these communities are huge for this organization. So this is something that is important for me, try to reduce the time of analysis, make this methodology available for everyone, community-based organization to governmental agencies. So this is a work in progress right now, and I think it's going to be a work in progress for the rest of my research career. You need to improve your method all the time, so to be able to get to that point. So basically, this is a methodology that I'm using that we developed in the lab. So collection, filtration, chemioxidation, clean, and then we use Raman analysis for our identification, detection, and quantification of the particle. So I'm not going to go through this very much. I think Gordon talked about Raman last week. For some of you that weren't here, Raman is a vibrational technique in which we use a source of energy, which is a laser, a monochromatic light, a laser, and that energy hits the molecules and atoms of our material. And those molecules and atoms are going to be vibrate in a specific way. This type of vibration, these are atoms. And that energy goes back to the detector of our Raman, and that detector can translate that energy into this beautiful spectrum. So these are specific peaks, which tell you what type of atoms and the bonds that they represent here. And with this, you are 100% sure that the material that you are analyzing, for example, in this case, is polyethylene. So this is the machine that we have here down the hallway. Again, if you want to go and visit. But how we apply Raman to our specific problem, because Raman have, I don't know, hundreds of applications. So how we apply Raman to our specific problem? Well, this is represent our filter. Remember, our filter has been filtered, our sample has been filtered, our filter is clean now. We can take it to the Raman, which is a microscope with a laser attached. So we put our filter in the microscope. And if you are familiar with counting bacteria under the infrared microscope, this is the same concept. So I was using something that I was familiar and applied that knowledge to a new question. So what I do is this is the surface of the filter. I select randomly 30 to 40 regions of interest of field of view of the microscope. And I analyze each of these areas looking for the microplastics. Okay, so these areas can change in size. So usually I do 100 by 100 micrometers. So give me an area of 10,000 square micrometers for each of these. So that produces 10,000 spectra. So the grid goes like this and like this. And what their equipment do is just go, this is the objective, the laser comes to the objective, this is my filter, and every time that the laser hits the surface, produces a spectrum like that. And I know what I'm seeing in that filter. So after I have all the area interrogate, I can produce this image, false color image, in which each color is assigned to a specific spectrum. So for example, this particle was a specific spectrum. All these red colors are this spectrum, so assign red to all these. So knowing this, I can detect the particles now, and I can calculate particles per liter. But not only that, I know that my grid is 1 micron by 1 micron. So I can calculate the area of this field. So now I have all my areas interrogate. I apply this equation, and I end with number of particles per liter of water. So now I have particles. I can calculate the area of each of those particles, and then I can calculate the equivalent spherical diameter, which is a diameter, assuming that that particle is a sphere, and it gives me a value. And with this diameter, I can calculate the volume of these particles. This comes with a lot of assumptions and a lot of uncertainties, but we take into account all those uncertainties in every step of our methodology, our protocol. So knowing the volume, and because the Raman tell me the identity of the polymer, I can now calculate the mass of the particles, which is density of the polymer, which is something that you can look on internet, multiplied by the volume of the particles. Now I have everything that I need. I have number of particles, I have area, I have volume, and I have the masses of those particles per liter of water in my sample. But not only that, I can do 3D image, 3D mapping of those particles if I'm interested in doing that. So what I do is the 2D mapping is exactly the same, but I can go up using layers. And now I can create a 3D image of that small particles. And that is important because, for example, these particles was around 500, 600 micrometer, and you can see different colors, and those different colors represent different polymers. So this particle was made of different polymers. We have polypropylene and polyethylene here. This is polypropylene, this is polyethylene. And then we have this pigment that we have found in every sample that we analyze. Every sample, sediment, water, and no matter where the water is from, we found this pigment. So we know these, for example, particles came from the recycle process because you can, and the industry don't, when they're doing new plastics, they don't mix polypropylene with polyethylene. Those polymers don't mix very well, and they produce low quality plastics. So we assume that this particle came from recycled process or a recycled product. So I'm going to give you 3 examples of what I'm doing right now using this methodology and other methodologies. The first one, the first example is analyze microplastic and blank carbon particles in the south shore of Long Island. So we want to apply our methodology to distinguish between these 2 particles in our water samples. This is a bright field image of our filter, where you see in the background is our filter. This is a microscope picture. And what you're seeing there, all the colors represent different materials in that field of view, in that region of interest. So what you're seeing here, the white is black carbon, the blue here is polypropylene, the red one is polyethylene, and then we have titanium oxide and quartz. So now we can distinguish between these particles. We know what is microplastic, we know what is black carbon. And this is important because, again, if you remember at the beginning, these particles are made of carbon. So we want to distinguish carbon from these 2 types of particles. So this work was to prove that we could do that. So, and of course, the size of the particles. So for this, we took advantage of a project that SoMAS was conducted back in 2017. They were doing water qualities around this area in the south shore of Long Island, and they were measuring the outflow, the water around the outflow discharge of the Southwest Sewer District. So you can see here, we're 9 station number 6 was on top of that pipe. So what it's a pipe that came from Long Island, from the island, and discharged the water from this sewer district here. So important thing about this, the size distribution of the particles, again, 95% is smaller than 5 micrometers, 99% is smaller than 13 micrometers. So very small particles. And you can see here the diversity of polymers, polystyrene, polypropylene, polyethylene, the pigment that I talked about it. And we found also Teflon in these samples. So I understand that Teflon is a very toxic product. And this map just shows you the distribution of the abundance and distribution of microplastics particles and mass. So you can see here, again, this is the outfall discharge. Red colors represent more particles, blue color, less particles, and the arrow represent the currents that are dominant in that area. So you can see more particles to the west of the outfall discharge. So what I believe is that this pipe is discharging the particles, and the currents are moving the particles toward New York City, toward New York Bay. So for the mass, you cannot see the same trend. We have these big particles of big particles, 20 micrometer. But anyway, if you remove these particles from the analysis, it doesn't give you a pattern anyway. But you can see here the mass in grams, micrograms per liter of the microplastic, reach around 60. And number of particles goes from 95, 100,000 particles is the maximum per liter of water. So for the black carbon, we did the same analysis. You cannot see the same trend. More particles are to the east of the outfall discharge. Bigger particles here. Importantly, that black carbon particles were 10 times higher in abundance than microplastics. So we found more black carbon particles. So after all of this, the new analysis that we did with these samples, we took all these spectra from black carbon. And remember that I told you that you see that picture at the beginning. Black carbon is a continuum of different sources of carbon. You can have wildfires, you can have gasoline, diesel, charcoal. So all these black carbon are very similar in the carbon content, but chemically they are somehow different. So we can see that in the Raman spectrum. So what we do, we use reference materials, charcoal, diesel, and gas. And we apply a PCA, a principal component analysis, which just arrange your samples how they're similar. If they're similar, they put it together, and if they're not similar, they put it away. So you see that charcoal, gasoline, and diesel are grouped together, all this spectrum. And the important thing here is all my samples, the spectrum from black carbon in my samples in the station are grouped together with the diesel. So that's telling me that potentially these Raman spectrum are very similar to the diesel, and that black carbon came from diesel. So if we see here, this is a picture or a map of all the traffic in that area in Long Island, the South Shore. And you can see this is just 1 year, and these are all traffic routes of ships. So in that area, we have cargo ship, we have cruise ship, we have fisheries ships. Sorry. And we have a lot of different ships that some of them use diesel for the source of power. So we think that this is an important background because you can now assign a source of that black carbon to a specific reference material. So this have implication in the isotopic composition of these particles, and I'm going to talk about a little bit next. So the second example that I want to talk about is wildfire smoke deposition. So you remember back last year, 2023, in June, this huge fire came from Quebec, Canada, and affected us here in Long Island. So you can remember this picture all over the internet. You see this was 12 or 2 p.m. in the afternoon, and the sky looks like that. So very, very dark. And of course, we need to blame someone. So we blame Canada for all these problems. So you see here the plume of the smoke. Right now, that is June 7th, the day that the fire or the smoke arrived here in Long Island. And air quality of that day was very unhealthy. So I was able, I had the opportunity to go out there that day and take samples. So I was at home and I was, God, we had this fire here, and I was black carbon, small black carbon, water, let me see. So I went out and I took sample here in this point. This is Long Island, this is the Long Beach area, this is the point here. And I took samples around here for 9 days, starting the day of the fire and 9 days, consecutive 9 days after the day of the fire. So I applied the same methodology that I'm using for microplastic. And this is what I found. So this is black carbon again. This is the Raman spectrum of black carbon, 2 peaks. So these are the 9 days, and these are the picture of those 9 days. This is the June 7th, the day of the fire, and you can see how the sky cleaned after that, very fast. So the first day, the second day, third day, and I have a peak here on black carbon up to 2 million particles per liter of black carbon that day. So I was at that moment, and I would say still not, I'm not an expert of wildfires and smoke and atmospheric composition. So I keep reading about this, and suddenly I found out that there is other component in the smoke that we look for. So the other component is iron. So this is the spectrum of iron. So I went back to my samples and I started looking for iron. And the result that I found around 5 million particles per liter iron that same day that the black carbon peak arrived. And again, reading more things come with those smokes. So also the nutrients and that kind of stuff. So I went back and I found this peak of this material that we don't know, I don't know what it is yet. I assume, or I have the feel, or looking in the literature, I think it's some type of inorganic salt, but we don't know what it is yet. But we found a peak of almost 12 million particles per liter, the same day of the other 2 peaks. So that is the second day of the fire arrived. So when you see the smoke, when you went out that day and your eyes were itchy and you have problem to breathe, you don't see anything in the water. Next day, when it was clear, better day to go out, you see all these peaks in the water. So I still don't know what to do with this. I'm trying to figure out a different approach to do this. Also looking for more stuff in my samples and see what else I can find there. And of course, the second day, June 8 was when I found all of this. And one of the questions that I want to answer is this: is pyrogenic carbon iron nutrient stimulate phytoplankton biomass? So these have been proved in other parts of the world, but the thing is, a lot of these papers, research, they use data from the atmosphere. They use whatever they can find in the atmosphere those days, and then they extrapolate to the water. The difference here is I have sample from the water, and I know that the second day was the peak, and I have concentration of those in the water, not in the atmosphere. And these are some of the questions that I would like to answer, especially for Long Island. And maybe you can think, well, Long Island have a lot of iron or a lot of minerals, but what implication have for low nutrient, low chlorophyll, high nutrient areas in the ocean when you have this input of iron, for example. And suddenly in one day you have this peak. So the last thing that I want to talk is another part of my work is how we demonstrate all of these things that I'm telling you about microplastic and black carbon in our measurement of the carbon cycle. So how we can demonstrate that? How we can demonstrate that indeed microplastic and black carbon are affecting our understanding of the carbon cycle in the ocean. How we can demonstrate the microplastic affect our instrument, our measurement. So for that, and first I need to address this. Microplastic particles can inadvertently analyze together with biogenic carbon in our sample. It's just because for analysis of different carbon pools in the water, we just take out a volume of water, we filter that volume of water, and then we use this equipment, elemental analysis, And this is equipment give you bulk characteristic of that water sample. It doesn't give you a specific characteristic of the, for example, the carbon that is in that water. It just gives you a number. This is the amount of carbon that is in this water. This is the amount of nitrogen that is in your sample. So you cannot distinguish between different types of carbon. Okay. So that is one of the things that we need to prove first. Is this type of equipment be biased by the presence of microplastics in our samples? So that's the first thing that we need to answer. So for that, I use sediment from Long Island Sound here. I add plastic in different amounts. Like I increase the amount of plastic in my sample from pure sediment to pure plastic. And then I compare direct observation of carbon percentage, carbon yields, predicted carbon yields between what I expecting using direct observation and expected values. So I use I had to thank Steve because he developed all the equations. So using mathematics, using conservation of mass, we know how to expect if I add certain amount of plastic to certain amount of sediment, I know what to expect from that equipment to tell me. I know that using mathematics. So the question is, the experimental results are gonna fit that expected values? So that's what we were trying to understand here. And if so, we have a problem because our equipment are measuring the carbon from plastic and you cannot distinguish that carbon. So you are assuming that all that carbon that came from your sample is biogenic, is particular organic carbon. And that is a problem. So I'm gonna give you this result here. This is two different polymers. The gray one is polystyrene, the black one is polyethylene. So measurement, so the values that the machine gave us is the circles. Least square regression is the dashed line, and our predicted relationship is the solid line. So our predicted relationship is what mathematics is telling us that's supposed to be there by conservation of mass. Anything that we put there, any atom of carbon should be at the end also. So that is what mathematics is telling us. And you can see here how our experimental result fit or fall on top of our predicted relationship. So that is telling us that that machine that we use is combusting all these carbon atoms and is producing this result. And we assume that all carbon is from biogenic material, in which we know now that is not true. So if you see here, when you increase the amount of plastic, of course, the carbon yield increase, that amount of carbon increase. So what you see here is from zero pure sediment to pure plastic. So the same thing, we test another polymer because we have only one polymer, what's happening with other polymers. So it's kind of the same, we have more scattered result here, but it falls very good with an R square of 0.8, our measurement with our predicted relationship. So starting with this, we know now that our equipment, our technology is measuring this. So we move forward and we wanted also to test not only the amount of carbon or nitrogen in those samples, we want to also test the isotopic composition of those plastics, how affect the isotopic composition of our sediment and our interpretation of that. And remember, isotopes are very important, for example, to understand the age of the carbon, how carbon circulate around the ocean currents. We use isotopic composition for a lot of stuff, and it's very important. So for this, we applied for the NOSAM research initiative. NOSAM is the National Ocean Science Accelerator Mass Spectrometry. This facility is in Woods Hole, in Massachusetts, Woods Hole Oceanographic Institute, Massachusetts. So if we apply for this grant, we were able to get there and they analyzed our samples. So again, the same concept, sediment with the inclusion of different mass of plastic from pure sediment to pure plastic. So we analyzed this in the mass spectrum in their AMS, and we had this result. So you have here from pure plastic, and then you add plastic to the sediment, and then you have pure sediment. So we have values of polyethylene's percentage carbon, the delta T13. For example, the age of the pure plastic is around 40,000 years. The age of the sediment was around 3,000 years. So we have all this information, and with all this information, we produce these results. So in what you see here in these plots are kind of the same that I showed you before. You have the mole fraction plastic carbon atoms. So this is the amount of carbon atoms that are in our samples that we add to those samples. And this is the delta 14 carbon measured. So this is what the equipment tells us, the give us, the experimental value. So again, we have the red, the solid line is our theoretical values, and you see how our experimental results fall with our theoretical expectations. So that's mean that this machine is measuring the isotope from this particle for this microplastic, and we can see this here. The same thing with the delta 13 carbon, exactly the same. You see here our experimental values, you see the theoretical line, and how they fit very well. So again, that tells us that that machine is measuring isotopic composition, and we don't know where this isotopic composition came from the sediment, from the plastic. If you have a sample, an unknown sample, we know now this here because we add the plastic there. So I want to show you this plot here. This is kind of a sensitive plot. So what we did here is, this is the carbon atom from polyethylene in percentage, 0 to 100. This is the mass, the percentage mass from polyethylene. So this is, if I take a piece of plastic and I put it in the sediment, piece of plastic, I don't care about carbon. I took a piece of plastic and I put it in my sediment. So this line represents different sediment with different percentage of carbon. For example, from 5%, this is hypothetically 5%, 2.5%, 1%, 0.5, and 0.1. So what's happening if I add a 1% by mass of plastic to my sediment sample? So you go here, this is 1%. And if we are analyze our sediment, 1% of carbon, we're gonna have 40% of those carbon that the machine is telling you that is biogenic carbon, 40% of those carbon came from polyethylene. 40%, almost half of the atoms of those carbons came from polyethylene, not from biogenic carbon. So your interpretation could change a lot for this. And that could be higher or lower. So using this relationship, for example, 1%, so now we know that 40% of this produce this, we can calculate the age error. And the age error, isotopic composition can tell you age of a material by C14. So we can calculate the error that potentially we can have if we introduce this. So 1% represent 1% of mass represent 40% of atoms. So we go here, 40% is 0.4 in our mole fraction. So if we measure this and we calculate the age error, it gives a 4,000 years error. And I'm gonna show you lastly what is important. And this is just showing you the amount, the blue square represents your sediment and how the amount of carbon from sediment is here, the amount of carbon from plastic is here. So I'm trying to show you how small had to be or can be the contamination of your samples in order to produce these huge errors. So how small had to be? If you work with sediment, it's almost impossible to detect this contamination in your sediment. So, and the smaller 0.5. So this is the part, this is the area that we're very interested in. This is small particle, this is small contamination. Because a big particle, you can think, a big part, I can see a big particle of plastic there. It's not gonna fit into my equipment, I'm gonna see the filter, but not those one. I'm interested in this type of contamination. So finally, I wanna show you this plot. This is like a summary of everything that I've been telling you. So let me try to explain the plot. We have here in the y-axis the delta 13 carbon and versus the carbon-nitrogen ratio. Carbon-nitrogen ratio is related to the red-field ratio, which gave a lot of information about the stoichiometry and relationship with nutrients and phytoplankton, for example. So we have this plot here. Each of these boxes represent common values of these two parameters. So marine POC, we expect having these values into this box. From the freshwater POC, we expect these values, freshwater DOC, marine VOC, and terrestrial material. So the black line here is our theoretical expectation. We calculate those numbers based on the amount of plastic that I put into the cell. So we calculate this. This is what's supposed to expect if we add 0%, which is this percentage, is the percentage contamination of plastic, 0%, 0.1, 0.3, 0.7, up to 8%. So that is the percent of plastic contamination in our sample. The black dots represent our experimental results. So these are the values of our equipment. And the pi and this number represent the potential source of those sediments, meaning terrestrial or marine. So let's start with this one here. And the red numbers represent the age of that sediment, of that sample. So let's start with the first one. The first one is our pure sediment. It fit very well into marine POC. That's what you expect for sediment. Fit in that box. Marine POC, it fit there. Okay, we have that, and it's telling us that we apply this simple mix model to try to understand the sources of the POC, of the organic matter. They come from terrestrial sources or marine sources. So we apply this model. Instead, the model tells us that 80% of that organic matter came from marine sources. 20% came from terrestrial sources. That makes sense. We have marine sediments, with the numbers here. But what's happened when we start adding plastic to these samples? So if we add 1%, again, a piece of plastic that represents 1% of your sediment or your sample can go up to, you can see how it moved from marine POC to basically terrestrial or marine POC or terrestrial sources. So you can see how the source, applying the model, how the source changed. Now it's telling you that 60% of that organic matter came from land, not from the ocean. And we know that it's not true because we know that our sediments came from the marine environment. So we can see here how the source assortment changed just when 1%. And not only when 1%, 0.7. And not only that, you can see the age that already showed you. The error was 4,000 years of error. Now, if you add this to this, you are going to have 7,000 years of age and old. So a huge error here in terms of the age of that carbon. In that age of that carbon, the interpretation can go wrong, for example, with dynamics of those sediments in that coastal area, how those sediments move around from terrestrial source to marine source, the mix of those sediments, how they mix each other. So think about the carbon pump in the ocean. All these things, all these things are used to measure all these processes. And if we don't have accurate numbers for these values, for these pools, we can have huge errors in our interpretation of all these processes. So finally, I just want to go through to significance. So this is what we were thinking back in 1990, no mention of microplastic or POC in the ocean at all. And then we moved to 2016, and we know now, 2024, that we proved experimentally that these things can affect our understanding, our budget, our numbers, our equipment. So it's time to make a pause and review what we're doing and think and see what we can improve our method, if we need to improve our method, and how we can address this problem. The other thing that I want to mention here is how microplastic and black carbon represent an important driver of climate change. So what I've been talking about is our numbers, our calculation, our interpretation, but how these things can actually affect climate change. So we know that black carbon is, and snow represent a very important factor for melting of the snow in the Arctic, for example, or in the Antarctic. So these particles absorb energy, and the snow surrounding these particles melt faster because these particles absorb energy from the sun. And we were clear about this. We know that black carbon is a huge driver for this, but now what's happening with all this microplastic that we are introducing to this environment? Microplastic have very similar optical characteristics with the black carbon, so they also tend to absorb energy, or they can't absorb energy. So they can potentially have the same effect of black carbon in the snow. The problem is, as I mentioned before, the method that we use only detects carbon. the method doesn't tell you what type of carbon are you analyzing. So using our methodology, we can address this problem. We can say, well, this is the amount of black carbon, this is the amount of microplastic in the snow. So we can point planes to black carbon and microplastic. You want to say like that. So this is something that I'm looking forward to address. Again, another aspect that I'm looking forward is just to try to understand how these black particles move from the surface of the ocean to deep water. We found these particles in the deep sediment, so possibly understand the marine snow, exopolymer particles, are potential transport vehicles from the microplastic to the ocean, to the deep ocean. Understand what type of microorganisms can degrade microplastic using, for example, a stable isotope probe, Raman, and fish. And try to see if we can do something about that. These are something that I've been working. And now we have this project that we are addressing distribution, identification, distribution source of microplastics in the Peconic Estuary. This is together with the Peconic Estuary Partnership funded by the New York State. We are doing this, but I want to span a little bit the project. So I went and I took samples in the Great South Bay and the Shinnecock Bay. So now I have sample, we have sample of these three big bays. So I want to see how these particles move around, how these particles behave, and how the distribution of these particles in this huge water body here in Long Island. So for those of you that are familiar with the Long Island, this is the Peconic Bay, this is the east end of Long Island, and Shinnecock and Great South Bay are also big bays here. Very huge environment. So I also want to mention very fast that I want to span this microplastic issue to human impact. So I've been talking to Dr. Jamie Milliker from Public Health Program to try to see if we can work with our methodology in human samples. So we are in conversation right now to do that. And also to see if we can address potential impacts in the marine food web for these small particles, and how zooplankton incorporate these particles into the food web, and potential implication for the seafood supply and human impacts. So that's it, and thank you very much.
Megan or Sarah?
I wanted to ask about the Raman spectroscopy method.
Yeah.
Why does it take so long? Is this the only method, or is it just the best one? And in which ways are you attempting to make the process faster?
Okay, so it takes so long because we need to interrogate a huge area of the filter. So we need to do all these small areas. And in that process, it takes time for the laser to act in the surface of the filter and process the analysis. So we're talking here that we call the time exposure of the laser to the surface. So you can say that it can be 10 minutes, 1 minute, where the laser actually touching the material. So you need to set up that. For that, I set up 0.7, trying to reduce the time. But again, it's around, I'm doing 10,000 spectrum for each area, so that take time. It's just time to take to do that. The other thing is there is several technologies to do that. For example, infrared Fourier transform, infrared FTIR is also used for this type, and it gives you the same kind of the same result. The only thing with infrared is you cannot go as smaller as with Raman. The threshold, the limit is a little bit higher. So, but there's different technologies that you can apply. And the way that I want to improve this is, for example, so right now I calculate different areas of the filter. So I go here randomly and I select an area, interrogate that area, and then I came with this part. But if you see the filter in the microscope, because of the contrast of the particles, you can distinguish particles from the background of the filter. So the only way that I have to do it is just go to the entire surface of the filter. So that's why I take 10,000. But imagine it's a technique that can show you the particles in whole filter. This is a particle, this is a particle, this is a particle. So you don't need to go all that. You can go directly to those particles and interrogate those particles. That is going to reduce the time a lot. So I'm planning to, or I'm working, I start working with the with dyes. For example, Nile red dye can attach to the plastic, and under fluorescence you can see those potential particles. And then just go directly to those particles. And use artificial intelligence. That is another thing that we're.
On a larger scale, how many sampling stations do you have and how are they spread out across the ocean?
Wow, yeah. I think I've been analyzed more than I have the Arctic, the Antarctic, the Caribbean, New York, a transfer from the Caribbean to New York. Now I have all these samples from the Great Salvation Echo. So basically the lab is full of bottles. I've been analyzed, I don't know, more than 200 samples, a lot of nights. But now I have to say, I spent a lot of nights during my PhD sleeping here, a lot of nights. But now, thank you to Tanya, we figured out a method to just let the equipment work the entire night. So I just had to set up the thing in the night and go through the night. So if someone want to work with me, I promise you don't have to sleep in the lab.
So it's a great presentation. I have a couple of questions. First about peroxide as a purification method for the plastics. I think I have seen some of the studies saying that peroxide potentially can alter some of the plastics as well. I just wanted to know whether you have any comments on that. And the second part about where you did the study on the wildfire smoke, the haze, very interesting study. And this is coming out of a little bit my ignorance as well, but why it takes 1 day for the particles, like the spikes to show up? And something interesting with the ion, like on day 9, it has spiked up. So I was just wondering like what's going on there.
Yeah, so the first thing is hydrogen peroxide. It's a huge body of literature now. With this issue, how you digest your sample, basically, how do you get rid of all these materials? So my decision is based off, so all of this is based to try to reduce contamination of your samples. So it's very easy to contaminate your sample when you're working with microplastics. So I'm pretty sure I'm just right now producing fibers here and things, because I know that, because I see my blanks, because I see the fibers that came from it. So my point was at the beginning was how I can reduce the possible contamination. So if you work with stronger or other digestive agent, like stronger acid, you need to take more steps to do all this kind of stuff. So every reagent have their potential problems with microplastics, and it was a matter of the result that I get right now. I'm getting very good results filtering, because that is another thing that I didn't mention. All these material that are in the water tend to clog the filter very fast, so you cannot filter that much water. And if you cannot filter that much water, you don't have a good representation of what's happening in the environment. So to reduce uncertainties, it's better if you filter more water. But with this 0.2 micron filters that I use, it tends to clog very fast. So I was able to filter 500 mL, in some cases 400. So with hydrogen peroxide, leaving that the sample digesting overnight at 60 degrees Celsius, the next day I can filter more than 1 liter. And it's very easy to apply. You don't have to, the potential contamination sources reduce. So I think for our methodology, I think it's the most convenient one, I would say.
Yeah, we're just wondering, like, have you done any control studies to look at how the peroxide may alter some of the constitution.
And the black carbon, I don't know why it takes 1 day for the signal to be present in the water. So those are one of the things that I want to understand. I'm not an expert in atmospheric things. I just saw that opportunity, black carbon, water. Let's see what's happening here. Let's answer these questions. So I don't know why it takes 1 day to deposit, but I think it's a very, in some of the paper, they mentioned that kind of stuff. How long it takes to. And again, the peak of iron and then day number 9, I don't know what is that. So I want to see. I had to mention I have more samples in the other bay. So the same days, but in other locations. So I need to analyze those to see what we have there. Thank you.
You said that some particles came from a recycling process because they had combinations. Can you speak to like what proportion of particles exhibit this recycling characteristic versus just pure plastic?
Yeah, we found a lot of these mixed particles. Like when I say mixed particles, it's different polymers in the same particles, polypropylene, polyethylene mixed together. And I would say it's like in every sample is like 2 or 3%. Of those, we found 2 or 3% of these particles in our samples, comparison with pure plastic. And with that pigment that I mentioned is on everywhere. And, you know,
So I just wondering, so how dissolvable microplastic will be and dissolving carbon in the ocean would drive the dissolution of the microplastics? Just curious about that. Because there are some like functional groups on the dissolved carbon. If there's chemical interactions between microplastic particles and dissolved carbon, I was thinking, would that be possible to drive the dissolution of the microplastics, potentially into nanoparts of plastics or some like dissolvable fractions of that?
so we know there is an interaction between microplastic and DOC.
Yes.
Is there like potentially that DOC can leachate from the plastic and that can be incorporated. Bacteria can use that DOC, and there is experiment in laboratory setup, experiment in the laboratory that have measured that how leachate from plastic can be incorporated by bacteria, and they can use that bacteria for growth. And that, of course, if you are losing carbon, of course you are losing part of your mass, and that makes the particles smaller through time. So that could be another source of fragmentation. If you want to talk about degradation of plastics, so leaching the carbon as a DOC with also particles and create smaller particles. So that could be a way of apply degradation by bacteria or interaction. So those are the things that we need to move forward. A lot of the things that have been proved right now is in the laboratory, like using bacteria from the lab, having incubation there. Okay, let's measure DOC, but what's happening in the environment. And we measure that in the environment also too, making incubation in the environment, that kind of stuff.
All right, thank you, Luis, for the nice.